Our last talk today is from Adam Simpkin at the University of Liverpool. He did his PhD there, finishing last year, in the group of Daniel Rigdon, looking at automated pipelines for molecular replacement in crystallography. He stayed on at Liverpool and continuing to work on crystallographic problems, but he took some time out to have a look at cryo-EM data, and I think he will tell us about this now. Okay, thank you, Adam.
Okay, thank you for that introduction. I'd also like to thank the organisers for inviting me to talk today. I'm going to be talking about MrBUMP and specifically the redeployment of automated search model generation for map fitting in CryoEM. So I'm going to start with a slide titled Who Am I? Because I expect many of you have no idea who I am. By day, I am funded by CCP-4 to develop software for molecular replacement pipelines. This includes SynBAD, AMPL, MrBUMP, which is the subject of this talk, and MRPARS. And I'm talking today because I saw a potential synergy between crystallography and cryo-EM that I found has yet to be widely explored. And so by night, I've been taking a little bit of time to explore MrBUMP's search model generation techniques. So an overview of this talk. I'm going to stray from the norm a bit and start my talk by not talking about cryo-EM at all and talking about some crystallography. But if we wade in, I will get back to applying how this is applicable to cryo-EM. I'm then going to give some preliminary results, an interesting example, and then some of the future directions I hope to explore. So, an introduction to molecular replacement. I'm taking the ill-advised start of saying X-rays are horrible, especially given that I'm still funded by CCP-4. But they're really difficult to work with in many ways. They sort of provide all the problems we have in crystallography. Instead of starting with an electron density post-data processing, we start with a diffraction pattern. And part of the problem with this is that we lose phase information and we need to regain this phase information. One of the strategies used to doing this is molecular replacement. And what molecular replacement does is it takes a crystal structure that is thought to be homologous and similar structurally to our target data and borrows the phases from this. We can see how this works if we put two very, very similar molecules or in the same orientation. And you can see that for each reflection, the phase information for this is very, very similar. And therefore, from a similar structure, we can sort of bootstrap a solution. Now, why am I telling you about this? Well, in molecular replacement, search models are critical. If we get a bad search model, we get bad phase estimates, and then we don't get any electron density. Therefore, programs like MrBUMP and AMPL have been developed to try and obtain the best possible search model using bioinformatic techniques. This can be a crystal structure that is sequentially similar and therefore likely to be structurally similar. This can be an ab initio model, and it's worth taking a little note here that ab initio models have got a lot better. That is, structures that are predicted just based on their sequence. This is one from Google's AlphaFold model. And finally, I just wanted to talk about a strategy that we use to try and get models to be more similar to our target. Here, I've taken this ab initio model and I've truncated it down, and this was actually necessary in order to solve the crystal structure. The final thing that I'm going to talk about is what actually is MrBUMP. Well, MrBUMP is a pipeline that goes through the entire molecular replacement strategy. It starts by examining the data. It then goes on to find a load of search models. It prepares these search models, and it performs molecular replacement for each search model before assessing the output solutions. The key advantage of MrBUMP is that it can automate all of these different steps, and this allows it to try and find the best possible search model available by running everything in parallel. Now, hopefully I can get this back to cryo-EM and why I think this relates. I can see that there are several parallels between crystallography and cryo-EM. I mean, if we look at these two very simplified flowcharts, we can see that there's similarities. We start by collecting data. We get an electron density map. we refine and do some model building, and we end up with a fully built structure. But there are also differences between the two. One of the things where I can see that is similar but not yet being exploited is that whilst we need to find a search model for molecular replacement, you also need to find a search model if you want to do model fitting and docking. What I'm trying to do is I'm just taking all the expertise that we've done in finding search models for molecular replacement and just sort of put this into the CryoEM pipeline. So some preliminary results. I selected GoEL as a test set. This was ideal as there are lots and lots of GoEL maps in the EMDB. There are lots of different resolutions. There's also quite a lot of other chaperonin proteins in the PDB that cover different sequence identities. It's a typical cryo-EM sort of problem, as there are 14 monomers in the structure, and the search model contained 3 domains with varied conformations. This makes it a little bit more of an interesting case, because if you take just sequence and you try and look for a homolog, you might find something that's in the wrong conformation whilst also 100% sequence identical. So my results so far, what I've got here is I've got a table of 11 different EM maps at various resolutions, and I've got 5 different structures that I use as search models at different sequence identities. If I have a look at these, they all look vaguely similar. I've scored these by Tempai's cross-correlation score, and what we can see is there's a sort of mosaic of some things that have worked and some things that haven't. This sort of works well with the sort of MrBUMP style of searching lots and lots of different structures at once, because there might be something that has that conformation but is sequentially less similar that can be caught in this way. This can be shown a little bit more obviously if we just focus on this 7 angstrom map. I had 3 solutions that gave sort of high CC scores and made me think, yeah, that's probably a solution. It's probably being placed correctly. The first with the highest CC score was perhaps unsurprisingly from the structure that was the most sequence identical. This placed 14 out of 14 structures, and the protein was mostly within the density. The next structure that I placed that had a high CC score was from the 50% sequence identity. And similarly, it looks really good. There's 14 out of 14 placed. It's mostly within the density. There are a few more errors, but it looks pretty good. The final one I placed had a 0.2 cc. It was just about looking like there may be things placed there. And only 9 out of the 14 were placed correctly, despite this having a higher sequence identity. This appeared to be a little bit conformationally different. And so the last 5 structures really struggled to pack into the density. So I decided that in order to try and get around this problem, I would go with a sort of crystallographics mainstay. I used ensemble to try and find the bits of the structures that were variable and just cut them off. And I thought this would make it more structurally similar and at the same time try and resolve some of the problems with packing. The truncated ensembles that I made look like this, and I think you'll probably agree these look very, very similar to one another. But when I actually tried to use these, they worked really, really well up to about 4 angstroms. And then beyond that, they didn't work very well at all. This seemed to counter the conventional wisdom in crystallography. Normally, we think it's much, much better to have something that's small and accurate than it is to have something that is larger. But here it seems that the long-range shape becomes more important. I'm going to describe some of the things that we can do in a little bit that may counter this. But first, I thought I'll go on and talk about an interesting, if possibly not unique, example. You see, I too have had a look at some SARS-CoV-2 proteins. This is the open form spike protein, and I used MrBUMP to find and prepare a search model for it. I used a program called Chainsaw that took an alignment between my search model and the sequence of my target, and then just truncated away anything that didn't match. One of the models that I picked out was the N-terminal domain from SARS-CoV, and this only had a 53% sequence identity. But nonetheless, 3 models were placed into the electron density, and you can see a little bit clearly that these aligned really well with the actual deposited structure. If I zoom in on this a little bit more, you can see that there are a few chains and strands and loops in here that don't seem to have been modeled in the original structure. If I look at this in Qt and put some density on it, the yellow model being the place domain, you can see that there are a few areas here where the model, there's a strand, particularly at the top here, that seems to fit really, really well into this density and yet wasn't built originally. And this is perhaps because it's something that would be difficult to just build when you're looking at residue by residue building. But using the place domain, you can see a little bit of additional information about the structure. You can see this being used maybe to guide model building or to be put into something like Redeployment to just try and automatically rebuild the structure. If you're looking at this structure and think it looks familiar, this is because this is the same map that Agnel modeled and talked about in his talk yesterday. This has been rebuilt and deposited into the coronavirus structural task force repository on GitHub. But he appeared to use a very, very similar method to this of placing a domain and then rebuilding. The nice thing about what we've done here is this has been fairly automated. I've not done anything particularly clever myself. And hopefully, if we can sort of make this method a little bit more easy to do, it can be done sort of more commonly. So finally, future directions that we wish to explore. So we can look at the map and we can look at the models. And when looking at the map, there are probably some very simple things to do that anyone who was looking at the nice symmetrical CryoEM model earlier was thinking, why aren't you cutting this up into individual segments and looking at these instead of trying to place all your models at once? That is something that we wish to do. I think being a little bit new to CryoEM, I didn't initially think to cut up the map, as normally my thoughts are just on how can I change my model. But this is another step that I think that we can do, and that would make things a lot easier. We can even potentially have a look at splitting our density into ways where we can have a look at individual domains. And this would certainly try and improve the problems that we're having with long range shape. The other thing we can have a look at is how can we change our models. And one of the ideas that we've had on this is to maybe use Balbus/Mortar style model generation. What these do is they take, in the case of this three-domain structure, they'll take the domains that are sequentially the most similar to the target, but then they will align them with many, many different homologs, which may have a range of different conformations. And then hopefully, by testing all these different conformations, they can find something that is more similar to their target. The other thing we can do is, when there aren't many homologs available, we could use a program like Concord, which sort of generates many, many different conformations for a structure. And by sort of creating an ensemble of different conformations and then clustering them, we can then just test these different conformations and hopefully maybe catch something that's unique and difficult. Another idea that we've sort of had, and I'm almost throwing out feelers on whether this is something that's really wanted, is looking at finding models for unknown density. In crystallography, I've written a program called Simbad that is used to find structurally similar search models when sequence-based searches fail. And this is a typical problem due to crystal contamination, but we see that maybe this is an idea that can be applied to unknown blobs of density that you see in your CryoEM maps. So if this is something that people are interested in, I'd be interested to hear back on this. So my acknowledgements, I'd like to thank Dan Rigdon, Ren Keegan and Martin for all their help in sort of doing these initial exploratory tests. I'd like to thank CCP-4 for funding me, and also University of Liverpool for, I was going to say housing me, but I'm housing me now. So without further ado, if you have any questions, I'll try and answer them as best I can.
Sorry, just finding my unmute button. Thank you very much, Adam. Okay, so we don't have so many questions this time. I think we're getting towards the end. So what is your view of using output from MrBUMP for molecular dynamics simulations?
I think that would be reasonable. If you could do a molecular dynamic simulation on your model, you might be able to find different conformations that might fit your density a little bit better. But it should be a fairly straightforward thing to do because MrBUMP is basically just trying to fit your model.
Yeah, okay. And another one, in a MrBUMP run, and typically how many models are you checking out? I mean, for crystallography as well as CryoEM, how many models are you typically checking to see if they fit?
I think this can depend on this is a bit of a case-by-case thing, depending on how many models can reasonably be found to in a sequence-based search. But I think you can do up to hundreds and hundreds of different models. It's as long as you want your computer to run, really.
Okay, and I mean, final one. I mean, how does your approach differ from using Chimera to rigid body fit?
So in this case, I've just been using MolRep to sort of fit the molecules into the density, but the way this is differing is on how we're finding these search models in the first place and how we're treating them. I want to say that this is where we're putting the focus on, in the fact that we're trying to find lots and lots of different search models, treat them in a way that we think will make them more likely to fit in the density. But the actual placement is maybe slightly different.
Okay. And final question, have you looked into using other similarity measures than cross-correlation when aligning the models to the maps?
Not currently. I just went with something simple and in the CCP-EM package when I just wanted to have an idea of how things were fitted. But I think that this is another thing that we can explore.
Yeah. Okay. Well, thank you for that. Yes, if you'd like to unshare that.
Oh, yeah, that would make sense.
That brings us to the end of today's session. So thanks to everyone for joining us.